Thank you. Well, it's really nice to be here this evening. And what's even better is that it's Fascination of Plants Day. So it's the perfect day for a plant science talk. I think, is that better? Okay. So what's even better about this is that it's Fascination of Plants Day, and I get to share with you my fascination with plants. And what I am really fascinated about is the diversity that you see in the plant kingdom with the shape that's there. And the shape can be really important for the function of the organs that you're looking at. And what's even more interesting to me is that all organs, be they a petal, a stamen, or a leaf, initiate from these dome-shaped structures, which is called a meristem, and it contains the stem cells, and stem cells can generate any type of cell. And organs form on the flanks of these meristems, and they're, again, kind of flattened dome shape, and they look absolutely nothing like the organ that they ultimately end up in. So what's going on in this process here during development when you go from this dome shape through to this complex shape that can be a 3D shape, and there's multiple different tissues in there. So I'm really interested in finding out what the rules are that control this patterning mechanism and how they have changed over time to generate a diverse set of shapes that have significant impacts on their function. So, for example, cup-shaped leaves like this pitcher plant are really important for carnivorous plants, but they're not so good at photosynthesis. So depending on what your function is, you need a different shape. So driven by this aim, my lab asks a series of questions. So what defines organ shape? How do new shapes arise? And how does shape impact function? And to do this, we use our favourite group of plants, and that's the grasses. And I know a lot of people aren't necessarily fans of grasses. Some have inherent hatred for it. And around this time of year, a lot of people are actually very frustrated with the grass because it keeps growing and you've got to keep mowing it. And that's what most people, when I talk to them about grass, think lawns. So why am I actually interested in them if they're such an annoying thing? Well, grasses are everywhere. They're a lot more than lawns. They're found in nearly every terrestrial biome on the planet, and they cover around half of the landmass. And they include, they're really important for many of our iconic biomes. So the savannas, the highlands have a lot of grasses in them as well. Anything in the rolling hills of England are all grasses as well. And they have really important roles providing food and shelter for animals and for us. But they also have really important roles engineering the soil structure. So this is even more important in areas like sand dunes, where we deliberately plant grasses to try and hold the sand dunes together. On top of that, we grow a wide variety of species of grasses for food. So this is an example of rice paddies. We have a lot of different species that we grow for our sports pitches. And we also have a lot of different ornamental varieties because they look beautiful in our gardens. So they're really important to us both environmentally and visually. But even more importantly, they actually comprise of over 70% of crop species, and 50% of global calories are directly provided by the cereal crops, which includes maize, wheat, rice, barley, and oats. They also provide luxury products like sugar and whiskey. And they feed many of our other food sources. So without grasses, we wouldn't have any dairy products or eggs or meat products. And increasingly, they're becoming really important for a sustainable source of biomass for industry. So grasses grow very quickly and they can produce a lot of biomass in a short period of time. So we're using them for building materials, making paper, and making biofuels. And for all of these different purposes, we're interested in different parts of the plant, and we're interested in different productivity levels. And so actually, they're so important to us. A world without grasses would be fairly catastrophic. And this has been thought about in the past. So this book, The Death of Grass, is about a virus that kills all grass species and civilization completely collapses. And it's not that far-fetched, considering they are in every aspect of our daily lives. But it wasn't that long ago that we didn't have any grasses. So grasses are fairly young in land plant evolution. They evolved around 65 million years ago, but they very, very quickly started to spread and became dominant in certain areas, generating the grasslands that we're very familiar with today. Nowadays, we have over 12,000 species of grass, and it's the 5th largest plant family. Even though people say grass is grass and it's boring, there is quite a lot of diversity, and especially in their floral structure. So this is the major area that we use to identify grasses. So how have they become so successful? What features are so important for them? Well, one theory comes back to the fact that grass keeps growing. So if you cut a grass seedling here, this is what you'll see over the next couple of days. So you can see rapid growth of new leaves. And this happens much, much faster than you would see if you cut, for example, a tree branch off. It's not going to produce its branches very quickly, but grasses can do this in a couple of days. And this is why they're so successful in a grazed environment, because they can be eaten and then they'll grow back and outcompete all the plants around them that aren't able to do this. And they're able to do this because of their special leaf structure. So if you look at a grass seedling, this is a maize seedling, it's nice and big, so that's why I use it. Most people I speak to point to this and say, that's the stem, I know what that is, but it's not. So as you start dissecting off leaves, you actually realize they're wrapped leaves. And these base of the leaf provides structural support and protection for all of these younger leaves, and the meristem cells are here. And the stem is about 1 millimeter in height at this stage. And what's important about this is that the meristem is now at or below soil level. It's buffered from sudden temperature changes, and it's protected from herbivores, because herbivores don't like a mouthful of soil. They're not going to take this out of the ground. They usually eat leafy bits at the top. And so the meristem is protected. It can continue to produce organs, and all of the young leaves that were already formed are ready to expand and come out. And that's why we're able to regrow so rapidly after grazing. So what is this leaf structure? It has this modular structure with a basal region called a sheath. It wraps completely around the stem and all of the younger tissues and provides strong vertical support to the plant. And this is responsible for the height gain that we see. Then you have this upper region called the blade that bends away from the plant and intercepts light, and it's the most important part for photosynthesis. And then the angle of the blade is controlled by the formation of specialized organs called the ligule and auricle at this boundary region. And if you don't have them, you have very upright leaves, and the plants aren't very good at producing grain, for example. So this angle is really important. And so this structure is responsible for potentially a part of the success of the grasses. And this is not just in maize. So if you look at seedlings across the grass family, they have different sizes and different leaf widths, but the same structure is there. This modular structure is always the same. So it's consistent across the grass family. But when you look outside of the grasses are part of this monocot grouping here. But the most common group that when I talk to people, like students, about what leaves are, they'll say, oh, these ones, they're flowering plants. They've got these leaves that have a narrow base and a wide upper region, but these are eudicot leaves. But this typical shape is actually seen in most of the other groups of flowering plants as well. So grasses are quite strange in their leaf structure, and they look quite different, because their ancestor is likely to actually have had this different leaf structure. So the most common leaf structure that if I asked you to draw a leaf, you would probably draw this, is the petiole base, which is a narrow kind of stem-like structure, and then a wide, roundish lamina, and sometimes there are these little leaf outgrowths at the base called stipules, and this is typical of a eudicot leaf. But the grass leaf looks very, very different, and we do not know how they relate to each other. There's been lots of theories, but we actually have no idea. We know a reasonable amount about how this kind of leaf structure forms because of model plants that we work with in the lab. But given how important grasses are, we know remarkably little about how the grass leaf develops. And so that's why I became fascinated in grass leaves. And so we're asking questions about how the grass grows. How does grass grow? What defines the leaf shape in grasses? How does this new leaf shape arise? And how does the grass leaf shape impact its function? Now, this last question is possibly the easier one because it's the one that people have been looking at the longest, so I haven't had to do any work on it. Leaf size and shape is developmentally controlled to minimize self-shading because the leaves are in an opposite fashion. So this is a mature maize plant. You can see that each leaf has a different angle, it has a different width, it has a different size, but the shape is still consistent. And this is so that the top leaves don't shade these lower leaves, so you can get maximal production to a single plant. So if you're growing maize in your back garden or in your allotment, this is the kind of shape of a plant that you would want. But when we're looking at large monoculture fields where we have very dense planting, this open leaf structure is actually detrimental. So when we're growing in these monocultures, what researchers have found is that you want a more upright leaf structure so that you shade your neighbours less. You get less productivity per plant, but because you can put more plants in the field, you get much higher yield. And so we're really interested in leaf structure and shape as an agronomic trait. And if we can understand more about it, we can use what we know to help guide breeding and precision engineering approaches to improve our crops. So we know that the leaf shape is important, but what actually defines the leaf shape and what can we use to modulate its shape? Well, leaf shape is important for function, but how do we define the leaf shape? This is the first step that we have in this process. So we can do some very simple things. We can measure leaf length. We can measure blade length, blade width, blade angle. So those are the things that we've done traditionally. And this gives us some really good data. But what we've found is it doesn't give us a comprehensive understanding of what the leaf shape is like. So we've moved on to doing some leaf scanning. So we use this wand scanner that you can just run along a detached leaf to get a full image. And these maize leaves are about 1 meter long, just the blade alone. So they're quite big. And if we scan these, we can then extract the shape. Do this for lots of different leaves, either for different plants or different leaves within a plant. And then we can use mathematical equations to precisely define the shape of the leaf. And this is an example here where we have a population with wild type and individuals that have a defect in a particular gene. And previously, when we did leaf length, width, angle measurements, there was no difference recorded in their leaf shape. But by taking this approach, we can actually see there's a very distinct difference in their shape overall. And these actually become much more rounded in their shape than the wild type. And so this tells us that there is actually something that this gene is involved in regulating leaf shape, and we didn't know about that before. And this is important if we can combine this with traits that we're interested in, like yield, because we can find out what specific shape differences are actually important and which ones we can ignore. But without knowing what the shapes are, we can't do that. So this is all I've told you about how we characterize the mature leaf shape, but as I said at the beginning, leaves initiate from meristems, which are domes. So this is a 3D image of a maize meristem. In blue are the meristem cells, and this green dotted line shows you the leaf primordium. And we wanted to look at it in 3D because maize leaves actually don't look like the meristem that I showed you right at the start. They don't have a dome shape. They actually form a ring to start off with, completely encircling the meristem. So they're completely different. They then grow up on one side, forming a hood-like shape. Then the margins start to expand here until they wrap around completely, and they completely enclose the meristem and the developing leaves. And this is really important for the heat protection of these leaves. So I have a line where I can heat shock them and see which cells get heat shocked or exposed to that heat temperature. And these meristem cells do not get heat shocked. They're so protected by so many layers of leaves that they're quite happy. They never see that shock. And then at later stages, we start to see expansion of the leaf, and eventually it emerges through the sheath, and you end up with the bending back of the blade. So we can refine this into 3 regions, 3 domains: initiation, wrapping, and expansion and wrapping phases. And then through looking at the 3D images, we can actually see that cell structures are changing at this stage, and the sheath and ligule regions can become completely distinct. At this stage in development, the leaf is 3 millimeters in height. At this stage, it's 1.5 meters in length. So there's a huge amount of growth and deformation that happens between these stages, but all of the tissues that are in the adult leaf are defined and set at this stage. That tells us that all of the patterning that we're interested in that defines the shape happens during these very, very early stages of leaf development, which are a lot harder to look at, unfortunately. Okay, so we know what the leaf does, we know how it changes over time, but how does it get these changes? And we know that genes are involved in regulating development. So what genes are involved in regulating the grass leaf development? And I'm really lucky because I work in maize. It's a fantastic genetic system. It's really easy to do crosses. So I get to go out to the USA every year and I spend the summer doing genetics experiments. So this is a video of my maize field last year. These here are wild type plants and they grow above my head. They're quite tall, very happy plants. And as you walk through the field, you walk along the rows and you come across your mutants. So this is a mutant we're working on at the moment. It's below my knee height, so it's very small. And this is what happens with a lot of leaf mutants. They're not very happy because you are affecting a really important aspect of the plant development. And by looking for these mutants, we can find various defects, and then we try and characterize what the defect is, what stage is it affected at, and what's the gene that's controlling that process. So here are some examples that we're working on at the moment. So fuse leaf 1 is a mutant that fails to separate the leaves. So instead of having nice wrapping leaves, they all end up growing together and you cannot unwrap the leaf sheets. Hoja loca means crazy leaf in Spanish, and that's because it forms tube leaves. So this is an adult plant in the field. It comes to about my hip height, and these are the younger leaves that are bursting out the side of a tube leaf because they cannot get out through the top of the plant. And then Truffula, we affectionately named it after the Dr. Seuss trees because in the severe mutants, we have this puff of leaves at the top of the plant. And it can produce up to 70 leaves when maize normally produces 18 very rigidly. So something has gone wrong with its planning and it does not know how many leaves it's meant to be generating. So by looking at what's happening in these mutants, we can find out a bit more about what's going on and what stages are the most important for defining final leaf shape. So Hoja loca is a really good example of this because we took these plants and we looked at their early leaf development and we found that they couldn't do this transition from a ring to a hood-like primordium. So this is the meristem of Hoja here, and then this is the leaf primordium, and it forms a ring and it stays as a ring. It never transitions through those shape changes that we characterize using our 3D imaging. So that tells us that the gene that is defective in Hoja is involved in making this differentiation between the middle of the leaf and the edges of the leaf. So we can start building up our gene networks and our understanding of leaf development by looking at these phenotypes. And by looking at lots of different mutants, both ones that we've cloned and that have been published, we can start building in a general map of gene identities across the leaf. So these, we may not know what the exact genes are. It may be hundreds of genes that are involved in these, but we can see from mutant phenotypes that there are distinct regions that have distinct behaviors. So this is an example from the middle to the edges of the leaf. There are 3 domains that we found. The central region in dark blue, this lateral region in red, and then this light blue marginal region. And we can map that back to the initiating leaf as well. So these are established right in the earliest stages of leaf initiation. So we know these regions have to be defined at this stage. When we're looking from the base to the tip of the leaf, this one's much easier because we can see this at the wild type plant, but we have the sheath region and the blade region have to be completely distinct. And we know when these are established during this stage of leaf development, when this leaf is about 3 millimeters in height. So by understanding when these identities are activated and how they're influencing shape, we can start to build a better understanding of what might be important if we want to engineer a new crop plant with better leaf shape. So we have our patterning genes, but how do they affect leaf shape? Well, they've got to have some effect on growth. We don't necessarily know how they connect with growth, but what is going on in the plant. And this has been something that's been next to impossible to understand in grass leaves because of this enclosed nature of the meristem. We can't actually watch it. So we've been working quite hard to develop a new method to be able to watch this in real time. So we now have a method where we can use a confocal microscope, which uses laser scanning to identify cells in the developing leaf primordium and track them over a period of time. So this is our first 0 time point, and we've been able to segment out every cell, so the different colours indicate a cell. 24 hours later, we take the same image and we segment again, and we can say, okay, so this orange cell here corresponds to this cell here, and it is growing by this much. In white dots, we can also say these cells have divided in these regions. And by mapping this information, we've been able to show that different regions of the leaf grow at different rates. And this is important for its leaf shape at the end. So we can define genes that are involved in leaf development. We can watch growth now, which is really nice. And we can define the leaf shape at the end. But what's going on in this region here? Growth doesn't automatically end up as shape. So how is this being translated into the final leaf shape? And this is a really important question because we could say, hey, we found a gene here. It's involved in the middle of the leaf forming. Let's engineer it and see if we get a better plant. What's likely to happen is we get a dead plant because we don't know how it's affecting the shape in the end. We don't know the methods or the processes that it's going through to generate a new shape. And so it's not predictive. So we need to know a lot more about this before we can start messing around with our plant architecture and our plant shape for crop improvement. And it's a difficult thing to do because we can't break down a grass plant to its finite components and then rebuild it in the lab. So what we do instead is we use computational modeling because we can build a digital plant from basics up and find out what is the most important parts. So we take the information that we have from our patterning, our genes that we know about, and we have growth rules. So for example, we know because plant cells have a cell wall, the cells can't slide past each other. So that is a rule that we have in our model. So we have a series of growth rules and a series of patterning rules that we have defined from our mutant analyses. And then we ask it to generate a model. And this is our most advanced model that we have. So we have our patterns in the different colors and we have different growth rules that we've introduced. And we can generate a leaf shape. We then take this model and we compare it back to the 3D images that we had generated before and say, how similar is it? If it's not very similar, we go back and we redo the model. We have new parameters. If it looks similar, we say we're done. So this model is able to capture the transition from a ring shape to a hood shape, to then wrapping and enclosing the meristem, to then expanding out to around this kind of stage in leaf development. Although it is a lot shorter for those of you who are keen-eyed, and that's because computationally we could not make a 1 meter size leaf. It has to be a much smaller one. But I can say it looks the same, but does it actually capture the behavior of real biological leaves? And one way we can test this is by looking at mutants. So this is a wild-type maize leaf with its leaf primordia successively, and then this is the base of the blade and the top of the sheath in a wild-type plant. This WOX mutant in maize is a classic mutant in maize because it has this very, very narrow blade and a very narrow sheath that's open, so you can actually see the internodes and the younger leaves inside. And if you track development back, you can see it doesn't do this enclosing stage, it remains open. We called it kind of a keyhole, you could see inside. But at this earlier stage, it looks remarkably similar. So it seems to be mostly affecting this later stage onwards. So we asked, could we replicate this mutant in our model? If we can, it tells us that our model is capturing the major elements of leaf development. If it can't, then we have to go back to the drawing board. So our wild-type model looks like this, and we hypothesized that this WOX gene controls the definition of this light blue region, so we took it away in the model. If we take it away, the first stage looks pretty similar, which is a good sign. The second stage remains open, it never wraps around, so it has this keyhole trait. And then in the mature model, we end up with this open sheath region and a very narrow blade, which does replicate what we see in the wax mutant biologically. So that suggests to us that we had captured many of the main elements of leaf development in our grass leaf model. So we have a model. What does it actually tell us about how grass leaves grow? Well, it tells us that gene expression in distinct regions of the leaf are essential for defining the final leaf shape, and that these are specified right at the earliest stages of leaf initiation. It's not that the leaf initiates and kind of grows and then new things come in slowly. It has to happen during these first stages when it's less than 3 millimeters in length. And then the other thing that it tells us is that different regions affect growth differently, and it changes over developmental time. So we can have a region that initially starts off constricting growth, later becomes a promotion of growth. What genes control this, we have no idea. They were just parameters that were in the model, and that's something that we now need to go and investigate to see whether or not our predictions are correct. And the major conclusion we actually have is that we haven't found all of the components yet. A lot of people would argue this isn't a grass leaf. It doesn't look exactly like a grass leaf. And that's partly because it looks much more like this unfolding stage, and it does not look like this mature stage. And that is because we haven't got in the model anything about this ligule and auricle region at all, because we know very little about it. So it's a work in progress, but it's giving us some predictions that we can go away and test, and good insight that we can find out whether or not the genes that we think are important are actually important, and what are they doing. So I've told you a bit about what defines leaf shape in grasses and how we're going about trying to find the rules that are important, but how do new shapes arise, and can we use what we have to assess this? So I told you right at the start that grasses look very different to many other plants. And so can we use our computational model to try and understand the relationship between them and work out how maybe they might have evolved? Unfortunately, there's a lot of debate about whether or not a grass leaf is homologous to a eudicot leaf. And this has been a debate since the 1850s, and no one has been able to really conclude anything solidly about it. Because if you can't say the petiole is homologous to the sheath, we can't translate any of the knowledge that we know from eudicots to grasses and back and see whether or not we can work out which genes were the ones that were important for the evolution of this novel shape. So currently there are 3 hypotheses in the literature. The first, proposed in 1850, is that the petiole is homologous to the sheath and the lamina to the blade. And this was just based on spatial distribution and some morphology observations. The second hypothesis was that the petiole is homologous to the entire grass leaf, and that's because the petiole has parallel venation and the grass leaf has parallel venation. And this hypothesis proposed that if there was a little bit of the lamina, it would be right at the tip and it dies during development. And then there was a third hypothesis proposed in the 1940s, that there is an upper leaf zone in green here and a lower leaf zone, and that the grass leaf is homologous to most of the grass leaf is homologous to the lower leaf zone, and a tiny bit is the upper leaf zone. And this was also based on the venation patterns. And the person that proposed this said, well, there's no evidence of the tip dying, so hypothesis 2 is completely incorrect. So thankfully, there was one hypothesis that was removed from our question, but we could not tell the difference between these 2 hypotheses. And actually, when we started this study, this was the predominant hypothesis that was accepted in the field. But we couldn't find any evidence of why it was the one that everyone accepted. So we said, could we use a computational model to address the question of homology and begin this process of understanding how the grass leaf might have evolved from an ancestor that had a petiole leaf? So we have the idea is that we could take our grass leaf model and we would create a model of the petiole leaf using either hypothesis 1 or hypothesis 2 theories. The first thing that we have to do is look at the initiation of the leaf. So in grasses, I told you that they're unusual in that they have their meristome is completely encircled by their leaf primordium. Whereas in eudicots, we have these little bumps that form. So the first thing that we had to do is actually make our primordium, which is these coloured regions, much smaller. So a eudicot leaf primordium is about a third of the width of a grass leaf primordium. But we kept all of our domain patterns exactly the same. So we still have the blue, red, and light blue domains. So our identities, or our genetic identities, are exactly the same in the model. We said, using this, can we create eudicot leaves? So our first hypothesis used exactly the same model with smaller primordium, but said that the sheath and the blade will affect growth differently. So when the sheath comes in here, it will restrict growth more than it does in the grass leaf model. And we can actually generate a eudicot leaf. And it even has little stipules as well. So that was quite nice. So what about hypothesis 2? Hypothesis 3. So this one's a little trickier because we don't actually have lower leaf zones and upper leaf zones in our original grass leaf model because we didn't need them. So we had to add these to the model. And to do this, we had to track the tip of our grass leaf model back to its initiation point, mark it, and then use the same parameters to mark the same region in our eudicot model. So this is the upper leaf zone here is shown in orange and purple in this model. So we then use these domains on top of the identities that were already there to generate a eudicot leaf model. And unsurprisingly, because it's modeling, we can make a eudicot leaf. So a lot of people said, well, you can make it any way you want that. That's not really showing us anything. But what it does tell us is that the simplest model supports hypothesis 1 because we don't need to add or lose any elements when we develop the grass leaf. So evolution tends to not take the most complex route in theory. And so if we were to evolve a grass leaf from a PCL leaf, using this one, we wouldn't have to lose any of the major regulators of leaf development, but we would have to lose 2 major regulators or 3 regulators of if we have this hypothesis. So it suggests that this one might be more likely. But can we test the models further, like we did with the grass leaf model, to see whether or not they're actually accurate or not? So luckily, eudicots also have WOX mutants, so we can use the same process that we did for our grass leaf model. So a wild-type Arabidopsis plant, which is a eudicot, has a leaf that looks like this. It has a round lamina and a narrow petiole, and has very, very tiny stipules right at the base. If you have a single WOX mutant, you lose the stipules, but the leaf looks exactly the same. If you combine mutations in multiple WOX genes, you end up with a very, very narrow leaf like this. So we asked, could our 2 eudicot models actually replicate this? And could we tell the difference between them? So hypothesis 1, we can generate a wild-type model. If we take away this blue domain, we lose our stipules. If we take away part of the red domain, we end up with a very narrow leaf. So that's really promising. It can replicate the biology that we see. What about hypothesis 3? We can generate our wild-type leaf, including the stipules. And if we remove the blue domain, we lose the stipules. But if we continue to remove these lateral domains, we still only lose the stipules. There's no narrowing of the leaf. To narrow this leaf, we would have to remove everything but the central domain, which would kill the plant in biological reality. So it's not able to actually replicate the behavior that we see in biology, and that suggests that our hypothesis 1 model is more accurate. So both our simplest model and the mutant phenotype support that hypothesis 1 from the 1850s is actually potentially the correct homology model. We then said, okay, well, that's using something that we based the models on, because we know about the WOX mutants, but what about something that we haven't based the models on? And what we turned to was a mutant called blade-on petiole, and as its name suggests, it doesn't have any petiole in Arabidopsis. So they have these very elongated leaves, but they have no petiole in the base. And we said, if hypothesis 1 is true, we would predict that the BOP genes that control petiole identity would only be expressed in the sheath region. Whereas if hypothesis 3 is correct, the BOP genes would only be expressed in the lower leaf zone of the eudicot, and they would be expressed throughout the grass leaf. Now, Arabidopsis leaves are less than a centimetre in length, and so it's really hard to see whether or not BOP gene expression is at the base or is it throughout the petiole at the stage that it's expressed, because there's 2 or 3 cells present. So we couldn't necessarily tell the difference between that, but maize is a lot bigger and it's a lot easier to see. So we looked at the BOP gene expression in maize, and this is what we saw. So in here we have the meristem here, and these are a section right through all of the successive leaf layers. And here is the ligule at the top of the sheath. And the dark staining is the BOP gene expression. And we never observe BOP gene expression above the ligule, which tells us that it's specific to the sheath. And that would support hypothesis 1. And whilst we were doing this work, there was a lab working in rice who generated a multiple BOP mutant, and they knocked out sheath development completely. So that tells us that the BOP genes control both petiole identity and sheath identity, and this supports hypothesis 1. So by comparing our models, we're able to address this long-standing debate in the literature about which part of the leaf is homologous to which part in the grass leaf. And this goes on further to be able to tell us that a common genetic pattern most likely underlies both grass leaf and eudicot leaf development. So there are core patterns that are in all flowering plant leaves. And it is the way these core regulators interact with the downstream growth regulators that changes the shape. And so you don't need to knock out a whole domain in the leaf and then generate a new one to be able to have a new leaf shape. You can simply just change the amount of expression or the amount it's affecting its downstream growth targets. And now this is what we're going on to try and find out: what is going on in these genetic networks. And in particular, we're really interested in this base tip patterning. So we know from the morphology that we have really strong domains between the base to the tip of the leaf. And we also know that these domains are really important for defining the differences between the eudicot leaf and the grass leaf. So there are some classic maize mutants that don't have ligules, and these are called ligulus 2 and ligulus 1. They have very upright leaves, but we have absolutely no idea what they do. So we found them, we said, yeah, we've got genes, and then we left them. No one knows what they do. So we're now trying to find out what genes do they regulate, how do they connect with the patterning mechanism that's going on in here? Because this ligule is a very sharp line, but it's defined when the leaf is 2 millimeters in height, but you don't see the ligule until it's several centimeters. So there's something has to maintain this patterning, and how is that happening? And then I skipped over the fact that they're part of the monocots, and monocots also have parallel venation, and they also have an ensheathing leaf base. And so how did you go from a eudicot leaf to a monocot leaf, which has an ensheathing base and has parallel venation, but does not have this classic grass leaf shape with the very tightly wrapped sheath that it extends and the ligule and auricle? And so we're looking to see if the model can actually replicate the monocot leaf shapes, and whether or not the genes that we know are important in both eudicot and grass leaf development are also conserved, and how do they differ at each stage, so we can slowly build up a picture of exactly what changes happened to evolve this grass leaf. So what have I told you about? I've told you about how we're trying to understand leaf shape in the grasses and the genes that are involved in it. We get to do some really interesting experiments out in the field, but also a lot of computational modeling work. We're also using computational modeling to be able to effectively reverse engineer evolution to try and find out how a leaf is built, and then how does it relate to ones that we've never been able to compare properly before. And all of this is really at the aim of not only understanding the diversity that we've got in nature, but also to be able to provide new targets for precision engineering of our crops to improve productivity, irrespective of what agronomic practice you're using. So can we get a super maize to grow in your allotment, but also a different variety that's going to be really good for monoculture? But mostly what I hope that I've been able to show you today is that grasses deserve our fascination, and that they're not just an annoying lawn that we have to ignore most of the year. And with that, I'd like to thank my lab. I have really been very lucky with some really fantastic undergrad honours students who have been absolutely amazing. None of this work would be possible without collaborators. So the Scanlon Lab and the Cohen Lab and the Hague Lab, where I did my postdoc, were really fundamental in the computational modelling that I've shown you. The Bartlett Lab host me every summer to go and do many genetics experiments. And the Estelle and Strada Lab I've been working with on the Truffalo mutant. And finally, if you would like to find out more about the plant science research that we're doing at the University of Edinburgh, we're doing a lot of work with outreach and science communication, so please follow us on our social media. We also have a blog on our website, so do go check it out, and I'm happy to take any questions.
Lineups are really hard to identify, so that was all lots of new stuff for me. So have we got any questions, please? So you first there.
Is there anything to suggest that stipules and oracles might be?
Can you just repeat the question?
Yes. So the question was, is there anything to suggest that stipules and oracles are homologous? I think that was a Kaplan theory, and based on what we have found, they cannot be. And actually, the Scanlon Lab has recently published a paper using single-cell RNA-seq to look at the identity of cells in a developing leaf, and they found that the ligule and auricle cells are more similar to sheath genetic identity than they are to the blade. So they've been able to show that they originate from sheath cells, but the stipules just don't exist in grasses. And it's partly because they have this ensheathing leaf base. I think if we were able to restrict growth at the base, we would be able to artificially generate some, but that's a very difficult thing to do in grasses.
Richard?
Yes, so grasses, as you know, are very, very nested within the monocles. And probably the majority of monocles don't really have a differentiated upper and lower part of the leaf at all. So has anyone looked at something like, I don't know, a daffodil or an orchid to see, what happens if we accept that the PTO and the sheath are analogous? Then how does it must have kind of re-evolved this difference. The ability to do it must have been there, but quite possibly it has an ancestor where the leaf wasn't differentiated. Sorry if you've got to repeat all.
Yeah, no, that's okay. That's exactly the whole thing of my new grant. So I'm about to start working on this. So you can actually see in this orchid here, you do have a delineation, and it's a delineation of an abscission zone. And my hypothesis is that this is sheath and this is blade, and the abscission zone is at the boundary between them. I think they all have a sheath and a blade, but they differentiate and grow to different extents. So I think you can generate, well, in the model, I can really reduce the vertical growth of the sheath, and I end up with a leaf that looks very much like an orchid leaf. So I think it's that the patternings is still there, it's just their growth rates are different depending on what species you're looking at. And I think for those species that don't have the grasses way of dealing with the fact that they've got a sheath that's very close, they have to have a very small sheath and then just bend immediately away from the plant, because otherwise they would have very upright leaves and wouldn't be able to photosynthesize.
So I think one of the key differences between hypothesis 1 and hypothesis 3 prior to a lot of molecular biology is based on the observation of parallel venation, right?
Yeah.
So for hypothesis 3, what you need is the extension of the PTO to continue to be the blade, just maintain parallelization, where you get diffuse veins would be at the center of the seed all the time. So for hypothesis 1 to be correct, that would suggest you need a lot of signal in the blade to stop it. Is that likely to be a single gene? I think that's going to be a multi-gene event to stop it.
That's an interesting question. So the question was the difference between, or the original hypotheses were based on venation patterns, and grasses still have parallel venation irrespective of which hypothesis you're using. So why is it, if hypothesis 1 is correct, why does the blade have parallel venation? So we played around with this in the model. We can't do real vein formation because it's not a 3D model, but we can effectively draw lines on it. And what we found was the way that the grass leaf grows, it automatically generates parallel venation, whereas the eudicot leaf has curved venation. So part of the way veins are formed is by forming dips in hormone signal, basically between veins. And so if you've got a vein that is forming a curve, you're going to have regions in the middle that are going to have a very low hormone signal, and that will initiate a new branch. So you end up with a reticulate pattern. Whereas if you've got very parallel veins, the distances are much narrower, and you end up with only veins going in the same direction. That's the theory.
So you end up, the eudicot leaf would have essentially curved veins, and then basically got infilling to give
And that's what we see when we look at vein formation in eudicots. Although I will say, this is not a model of vein development, and there's a lot of debate about how veins form, and I'm not moving into that area.
Are the nodes homologous?
That's a really good question. And I had a debate with a colleague about this recently. So are nodes homologous between eudicots and grasses? Yeah. The theory is that flowering plants have this phytomer unit that is node, internode, organ, and axillary buds. You can see that really clearly in grasses, really nicely, and you can see that really clearly when you're looking at the flowering stages of eudicots, especially Arabidopsis. But there is some data from barley that suggests that they have effectively like a bare node internode pattern, and the leaf actually comes at the top. I don't necessarily believe that, but there's a lot of debate about it, so we don't actually know. This is one of the first studies where we've actually directly compared between the two. And so it's something that we're really interested in looking at. And actually, Sandy Hetherington and I are looking at how can we find out how things like nodes and phyllotaxy and things like that have different, are able to actually be compared between the two. But at the moment, it's unknown.
Right at the beginning of your talk, you told us about certain rules that you have to program to make the thing work. When you presented it, it seemed there were just a small number of rules. There must have been hundreds. So can you tell us how many rules you have to program to make it work?
Yeah, so the question is, how many rules do you actually need in a computational model to make it work? The answer is a lot. But it's mostly the principles are a lot fewer. So we have a lot of parameters that we can change, but the major rules there's not very many. So rules like cells can't slide past each other, that they are elastically connected. Those are all set in the model, and we don't change any of that information at all. What we do change is the equations that say this colored region affects growth in this way. I can tell you that we have close to 30 parameters for the grass leaf model, and we have slightly fewer for the eudicot model, so it's a bit simpler. But the principles are comparable between them.
And you don't have to have anything like gradients of hormones and water and things like that.
We do. Yeah, so I simplified it a lot, but we do have to have gradients of things across the tissue. So we have some, so for example, the patterns that I showed you, those are set domains that we can say this region has this color identity. But we also have some factors that are diffusible through the tissue to set domains in some cases and to affect growth in other cases. And we think they're homologous to hormone gradients, but actually we don't know any gradients in the grass leaf. So the tools just aren't there for us to be able to assess it quantitatively. And so that's something that we're now moving on to try and actually get that quantitative data and see whether or not our model could be completely wrong. But we don't know unless we look for the data now.
I just check my understanding. Say, if there is a large, what I would call stem with a fluorescence control, and a few nodes, where about, in this case, is the growth point? Where about it becomes a stem?
Okay, so the question is, if you have a large inflorescence stem with a few nodes, where is the meristem? So in vegetative growth, you have the stem and the meristem on top. The meristem itself produces both the stem and the organs. When that meristem transitions to flowering, and it will transition to make an inflorescence, it no longer produces stem. The stem internodes expand, and you see the inflorescence meristem emerge. If an animal eats that, the plant no longer has a meristem. But during vegetative growth, it is protected, and the stem nodes don't expand.
The meristem is still basically at the bottom. It's just not functioning.
No, it's always at the top of the stem.
It will be.
Yeah, it's just that it's transitioned to a different identity, and it's producing the inflorescence.
Going back to the one more.
Yeah.
I'm very scared of this about myself. If you, it's a more general question. If you cut grass, does that encourage it to grow faster or slower, or just stay the same? Keep growing.
Okay, so the question is, if you cut grass, do you encourage it to grow faster? I don't know. I think what you do is that you discourage non-grass plants around it. So that's why if you've got like sycamore seedlings, if you're mowing your lawn, that actually stops the sycamore seedlings establishing. But I haven't actually tested to see if it grows faster, so I don't know. That's a good question. So the question is, are the mutants naturally occurring, or have we done gene editing? So the maize mutant collection is fantastic. There are some that have just naturally occurred, and we found them in fields. Many we use a chemical called EMS, which causes point mutations in the DNA. It's pretty nasty for us to handle, but it causes point mutations in the DNA, and then we screen that mutant population. Increasingly with CRISPR editing, we're now able to target certain genes, but we're still at the stage that we don't really know what the effect of that would be. So it's more shooting in the dark for that process. So we tend to do these big mutant screens and look for things we're interested in at the moment.
The last question then.
Idea what an ideal leaf shape would look like. It says in the beginning towards or far end the plant that change the leaf shape that are typically.
So the question is, do we know what an ideal leaf shape is, and are we aiming for it, and do we have any mutants that are better? Generally, all of our mutants are miserable because we're really good at breaking things. We're really bad at making them better through mutagenesis. One of the things that we, or I keep running into actually, is in the maize field, we're really interested in leaf development, but in barley and wheat, it's been largely ignored. So we don't know what the ideal architecture is in those plants. And so actually we need to do that research to find it. Interestingly, we have a lot of novel grass crops that we're interested in bringing into the British agriculture system, but they haven't had this intensive breeding that barley, wheat, maize, rice have had. And so we actually could use what we know about the model systems that we have to rapidly engineer those to look better. So if we, for example, if we're interested in growing teff in the UK, does that mean that we need more upright leaves? We can target those and generate those plants fairly quickly now.
Okay, well, thank you very much, and thank you for all your questions. That was really good. And thank you.